this video, we will study about the genetics. What is genetics? Introduction to the genetics, various definition and history related to the genetics and its development. So let us start with the definitions. So first of all, before starting genetics, we have to have the basic understanding about the cytology. Cytology is, as the name suggesting, cytology is made up of two Greek words. One is the kaitos, that means the hollow vessel, and cell is the logos, means to do discourse, or simply we can say the cell biology. Simply the study of cell and its structure is known as cytology. So cell biology is the biological science which deals with the study of structure, function and molecular organization, growth, reproduction and genetics of cell. Okay. So if anybody asks about the cytology, cytology includes the structure of cell, the function of various organelles present in it, molecular level study and functioning of an organization of the cell, and the growth and the division of cell, or we can say the genetics of the cell. Okay, so this is the cytology. Then coming to the genetics, genetics is the biological sciences which deals with the mechanism of heredity and causes of variation in a living being. Okay, so genetics deals with the study of heredity and variation. What is heredity? Transmission of a character from parents to offspring is known as heredity. Okay, so the character which transferred from one generation to the next are known as heredity. And while the transfer of character, there are some variation. Those variation, their causes is studied inside the genetics. Okay, so the word genetics was derived from the Greek root, which means to become or to grow. It was coined by the Bateson in 1906. Then is the cytogenetics. Cytogenetics is the branch of genetics that is correlated with the structure, number, and behavior of specifically chromosomes with the heredity and variation which present inside the nucleus. So cytogenetics means the specialized segment of the cytology which study about only the genetics, or we can say the structure and the behavior of chromosome. Then as we study the genetics is the science that deals with the mechanism of heredity and the variation. Heredity means the study of transmission of character from one generation to the another. Therefore, it is fixed for a particular individual. Or simply we can say the transfer of character from parents to its offspring is known as heredity. Then what is inheritance? How it is different from the heredity? So inheritance is the trait which if transferred from parents to offspring are generally known as inherited trait, and the process is known as inheritance. So simply this mechanism, or we can say this whole process is known as process of inheritance. Heredity include both inheritance as well as the causes of variation. Inheritance are quite synonymous to each other. Difference among the individual of an species for a character is known as variation for that particular character, and it may be of two types. One is the hereditary variation. Again, heredity means when the trait is transferred from parent to offspring, and here we are depending on what at the genetic level, or we can say at the genic level or at chromosome level. So these are the variation at the genetic level. Such variation are found not only in the progeny of the different parent but also among the progeny of same parents. For example, for one pair of parent, if they produce three different offspring, so for a single trait, for example, height is a trait, so height will be differ in three of its progeny. So we cannot say that the trait will be fixed for an individual or for a parent. Okay, so the variations are there at the different parent, also among the progeny of the same parent. So for example, difference in the pattern of stripes in zebra and differences of length of neck in giraffe, or we can say all the characters behave in a similar manner. Then is the environmental variation. It is due to the impact of an environment on an individual which may affect the expression of genes. For example, difference in skin color, similarly different level of irrigation and fertilizer that lead to the different type of phenotype in the same variety. Means in the same population, if we give the different type of food and nutrition, the growth will be different. So this comes under the environmental variation. Then now coming to the history of genetics. So it starts with the Van Leeuwenhoek. Van Leeuwenhoek is recognized as the father of microbiology. Why? Because he was the first to observe the bacteria and protozoa with his improved lens system of microscope, which he made by the grinding. Okay, then second is the Linnaeus. Carolus Linnaeus is considered as the father of taxonomy. We are well aware of it. that he developed a way of naming and organize a species that we still use today. Like for an example, we say that it is Triticum aestivum. Okay, so Triticum is genera and aestivum is species. So we are still following this naming system. So what he gave us, first is the hierarchical classification system and second one is the binomial nomenclature. It's a two-part naming method. In 1735, he also published his first edition of Systema Naturae, which was the small pamphlet explaining the new system of classification of Naturae. It is very well famous book. Then is the Lamarck, 1744 to 1829. Lamarck is best known for his theory of inheritance of acquired characteristics. So basically, theory of inheritance came from this point. Okay, so he first presented in 1801 Darwin's first book dealing with the natural selection. It was published in 1859. So you can see the difference. Your natural selection book came in 1859 and Lamarck presented his theory in 1801. He said if an organism changes during life in order to adapt to an environment, those changes are passed into the offspring. Means if there are some phenotypic changes in the parent, that will also transfer to the progeny, although this theory is not accepted. Charles Darwin in 1809 to 1882, he put forth a very famous theory of the natural selection. He was the first who formulated Darwin book on the origin of species in 1859. It is the process by which organisms change over a time as a result of changes in heritable physical or behavioral traits. Like Lamarck said only about the physical traits, he also said that over a time as a result of change in heritable physical or behavioral traits. So he didn't talk only about the phenotype but also about the heritable changes. This theory is sometimes also described as the survival of fittest, but this can be misleading. Bobnier said here the fitness refers not only to the organism strength or athletic ability, but rather ability to survive and produce. So we cannot say only about the phenotype, also about the genetic ability of that particular character. In 1838, M.J. Sheridan and T. Schwann, they formulated the cell theory in plants and animals. This is the very well famous theory of cell. It states that all forms of life are made up of cells, whether animal or plants, and all the living organisms grow and reproduce because of the ability of cell to divide. So this is the two important point of cell theory, that all the living organism or form of life are made up of cell, and cell has the ability to divide. That's why all living things are growing and reproducing. Okay, so this is the well famous theory of cell. Then G.J. Mendel, father of genetics, 1822 to 1884. So father of genetics, G.J. Mendel done his experiments with pea plant. Then he published his result in Natural History Society of Lund at two of its meetings on February 8 and March 8, 1865. The paper Experiment in Plant Hybridization was presented in German language. At that time, nobody can understand that other than Germany. So it was published in the annual proceeding of the society, and it is totally presented in the statistical form. So again, scientists are also not able to understand what he presented. So at that time, nobody recognized his work, but in the year 1900, rediscovered botanist Hugo de Verais in Holland, Carl Erik Collins in Germany, and Erik von Teschmark in Austria were doing the work independently, and they concluded the work of Mendel simultaneously in the year 1900. So the work of Mendel, which was completed in 1865, was recognized in the year 1900 after the death of G.J. Mendel. And by that time, nobody knew that G.J. Mendel was the father of genetics. After this rediscovery, he was known as father of genetics. 1906, W.L. Johansen coined the term gene, genotype and phenotype, because 1900, after the law of genetics came into the light, the systemic research on biology related to the genetic came into a path. Okay, so what is gene? Gene is the basic physical and the functional unit of heredity, and genes are made up of DNA. Some genes act as an instruction to make a molecule called proteins. However, genes do not code for protein. So this is the very brief theory of gene. However, we will study in the coming lesson of genetics what is gene and how it is different from the allele and how they work and how they express themselves and independently. And coming to the genotype, the combination of allele that an individual possesses for a specific gene is known as genotype. Phenotype is The sum of organism observable characteristics means the physical expression of a character is known as phenotype. Genotype is the combination of alleles which is present in the chromosome or in the nucleus, and phenotype is what which we can see from our naked eyes. Genotype we can't see from our naked eyes. So genotype inherited from one generation to another, while phenotype do not. Genotype will be inherited because of the cell division, but phenotype will not. Then is the dominance. Dominance means in the heterozygote condition. Heterozygote condition, for example, there is a character height. For that height, it may be tall or it may be dwarf. So if you take the example of tall and dwarf, tall will be denoted by capital T and it is dominant one, and dwarf is the recessive allele and it is denominated by small t and it is the recessive. Heterozygote condition means if an individual having this genotype, and it will be tall definitely because of the dominance of this allele over this. So in this case, one allele express itself by suppressing the effect of another. This is known as dominance. So this allele is expressing himself by suppressing the effect of recessive one. So this is known as dominance. Similarly, recessiveness. This is the case of recessiveness, that in the heterozygotic condition, one allele which unable to express itself known as recessive allele. So for a heterozygotic, we can say the one allele which can express itself is known as dominant one, and which cannot express itself is known as recessive one. And how we can say that they are expressing? Based on the phenotype. Codominance, when both the allele of the gene express equally in the heterozygote, that condition is known as codominance. For example, AB blood group. 1909, Madsen coined the term epistasis. Epistasis is important for us because it is the exception to the law of independent assortment which was given by the G.J. Mendel. So epistasis means when an inheritance of one gene is affected by another, and this gene interaction is known as epistasis. These are the exceptions to the law of independent assortment. The phenotypic ratio combination of law of independent assortment, or we can say that dihybrid cross, is 9:3:3:1, but after epistasis, due to their interaction, it may be differed to the 15:1 or 13:3:1, or maybe in other combination. There are 6 other type of epistasis and we will study it in coming lessons. Okay, in 1909, Carl Correns reported the cytoplasmic inheritance in Mirabilis jalapa. Cytoplasmic inheritance means earlier and now up to we are studying about the nuclear inheritance, means genes which are present in a nucleus inside the chromosomes, and we are studying the inheritance of this thing. And Carl Correns in 1909 reported that apart from the nucleus, there are some organelles present in the cell which are able to inherit independently of nucleus. So what cytoplasmic inheritance said, that the transmission of genes that occur outside the nucleus, and it is found in most eukaryote, and it is commonly known to occur in cytoplasmic organelles such as mitochondria and chloroplast. These two autonomous organelles have their own genetic material. So apart from the nucleus, there are two other organelles which contains the separate genetic material. One is the mitochondria and chloroplast. Chloroplast found only in plants. 1910, T.H. Morgan studied the crossing over and the recombination in the Drosophila and coined the term crossing over. And you all know that the crossing over is the very important feature of meiosis. Crossing over is the exchange of genetic material between the non-sister chromatid of the homologous chromosome during the meiosis, which results in the new allelic combination in the daughter cell. That's why the variations occur at genetic level, and this is the very basic reason of variation during the transmission of character. In 1944, Avery, McCarty and MacLeod explained the significance of DNA and proved that this is the genetic material. And the experimental proof was DNA as a genetic material was given by the Hershey and Chase in the year 1952. 1953, Watson, Crick and Wilkins proposed the double helix model of DNA, and for the same they got the Nobel Prize. 1958, Beadle and Totem and Lederberg put forth the one gene, one enzyme theory, and this theory is very important for the genetic expression understanding. One gene and one enzyme hypothesis said there is one gene which is responsible for one enzyme, and that determines the phenotype of an character. 1958, Crick explained the central dogma of the molecular biology. From the 1958, biotechnology is also evolved. So He explained the central dogma of the microbiology. According to the central dogma, DNA to the RNA to the protein. So this is the unidirectional process, and this is known as central dogma. 1958, Measles and Install proved the experimentally that DNA replicates by the semi-conservative mechanism. So DNA replication model was given by the Measles and Install in 1958. 1961, Jacob and Monod explained the genetic regulatory mechanism in protein synthesis, or you can say the mechanism of the protein synthesis, which we widely known as operon concept. Lac operon concept is given by Jacob and Monod in 1961. In 1968, Narenberg, Khurana and Holloway deciphered the genetic code and the polynucleotide synthesis. They explained that genetic codes are responsible for the polynucleotide synthesis, ultimately leads to the phenotype of an character. Okay, so for the same they also got the Nobel Prize. 1975, Delvico, Tamin and Baltimore discovered the mechanism of the reverse transcription, which is known as Taminism. So what is reverse transcription? That is the synthesis of DNA from an RNA. As what we have studied in the central dogma, that DNA leads to the formation of RNA that then to protein. So but reverse transcription means RNA to DNA can be formed by the use of retroviruses. In the 1986, Gary Mullis developed the polymerase chain reaction technique, although this year is very different in different books. So as the year are not very much important, but you must have the knowledge of the scientist and mean what they did. So polymerase chain reaction is a very landmark achievement in case of molecular biology. The polymerase chain reaction is a technique which is used to make a numerous copy of the specific segment of DNA quickly and accurately. Means polymerase chain reaction is responsible for the amplification of DNA. So amplification of DNA can be done outside the cell with the help of polymerase chain reaction. So PCR enables investigator to obtain the large quantity of DNA that are required for various experiments, in the plant also, procedure in the molecular biology, forensic biology, and medical diagnostics. So PCR has very wide application nowadays. So this is about all the important historical events of the genetics, and for the same and for the reference you can study PDF where the detailed history was given. Thank you everyone. Hello everyone, in this video we'll study about the other genetic concept. Now first of all you have to understand that why we are saying this the other genetic concept. These are the other genetic concepts or the exceptions to the Mendel's law. These concepts are the non-Mendelian genetic concepts because they are not behaving according to the law of genetics. So to understand this particular video you should be well aware about the Mendel's law, or we can say the law of genetics according to the Mendel. Okay, so must watch the previous video before studying this particular topic. Okay, now we'll start. First we'll understand some of the exceptions to the Mendel law, then we'll discuss each and every exception in a detail. Okay, so the first and foremost is the paramutation and polyploidy. Paramutation and polyploidy. Paramutation and polyploidy is exception to the law of segregation or law of purity of gametes. Now, you know, law of segregation and law of purity of gametes are given by the Mendel. So these are the Mendel's law, but paramutation and polyploidy are not following these laws. Why? What we are saying in the law of segregation? We are saying that for two allele, despite of remaining together, these are the two allele for the gene height. Capital T is donating the tallness, small t is donating the dwarfness. So despite of remaining together in an individual, when the individual will form the gamete, so these two will segregate from each other without contaminating. They will segregate and they will not contaminate each other. Means despite of remaining together, these gametes are always pure. Means capital T will have its own effect whether it is present in this condition or in this condition. There will be no contamination. That's why we are calling it as a purity of gametes. This is the ideal condition while the gamete formations takes place. But due to permutations or in the polyploidy, what condition arises? So this is the individual condition, but for the gamete formation this should be the ideal condition. But what is happening? Despite of passing individually to each gamete, both the alleles are assorting in a single gamete, and this gamete
do not have any of the allele, so we are denoting it with the 0. So now assume this is a proper individual and this is the gamete by the individual having this kind of effect. Now what will happen when they will combine capital T and small t? So this is the condition of polyploidy, or this is the condition of 3n, but what should be the condition? 2n. 2n should be the condition. Despite of 2n, this individual is having the 3n condition, not viable. Again, capital T and no means no allele present here. So capital T and 0, this is the condition of an aneuploidy, and what should be the ideal condition? 2n. Again, this is not viable. Now again, capital T, small t, again this is polyploidy, and this is a small t and there is no allele. So this will again will become the hemizygous condition. So these are the exceptions and these are the non-viable forms. So these are the permutation and polyploidy which are exceptions to the Mendel's law of segregation. Okay, so this you have to remember. Now the second thing is linkage. Linkage is the exception to the Mendel's law of independent assortment. So the Mendel's law of independent assortment says that despite of remaining together, two or more character will assort independently of each other when an individual form the gamete. Now it is just similar to the law of segregation. In the law of segregation we were talking only about a single character. Here we are talking about the two character, like seed shape and the seed or the plant height. Okay, so according to the law of independent assortment, the alleles of one character will assort independently of this character. Means there is no obligation that dominant will always go with the dominant or dominant will go with the recessive. So there is no such kind of rule. There may be an equal chance, like capital R may go with the capital T along with the other allele combination. So these are the combination they may form with for other characters also. So this is the condition of independent assortment. But in the linkage, what is happening? In the linkage, see, these are the gamete formation. In the linkage, we are saying that few genes are very closely linked, or which are very closely in a particular chromosome, and they are assorting as a single unit in the next generation. So that is a linkage. Although linkage is good for plant breeding in some cases to understand the inheritance, but again this is a exception. Then is the incomplete dominance. Now what is incomplete dominance? Incomplete dominance is exception to the law of or principle of dominance. Now what principle of dominance says that? Principle of dominance says that homozygote will always be equal to the heterozygotic condition for a dominant allele. Like this individual will be tall having the capital T, and this individual having the capital T small t will also be tall because of the presence of this dominant allele. But incomplete dominance is exception to this condition. Means what we are saying, heterozygote will show the same impact as of homozygotic. Heterozygote will also will show the same impact as well as similar to the homozygote. But what is happening in the Mirabilis jalapov, that is 4 o'clock plant? These are the red flower. Capital R is denoting for the red flower. So capital R, small r, this should be red. But instead of having red kind of flower, plants having the capital R, small r genetic combination showing the pink flowers. So this is, and these are the white. Okay, so these are not showing the complete dominance effect. So that's why we are calling it incomplete dominance. So these are the exceptions. Now next one is the pleiotropism. This is exception to the principle of unit character. We generally say that one gene governs one character, but there are the characters, more than one characters are governed by the single gene. This is the case of pleiotropy. Then the modification of the F2 ratio, like for the monohybrid cross. In the monohybrid cross, the phenotypic ratio is 3:1, and in the dihybrid cross, the phenotypic ratio for the F2 generation is 9:3:3:1. But there are some conditions like incomplete dominance, codominance. Incomplete dominance lead to the modification of 1:2:1. This is the phenotypic ratio of the incomplete dominance for a monohybrid cross. So the lethal factor, interaction of factor, epistasis, and all these exceptions are there which are modifying this kind of ratio. So these are the exceptions to the Mendel law, or this we study in the what we can say in the other genetic concept. However, Mendel was failed in showing the result similar to the P in case of Hieracium, that is hawkweed. Why? Because in the hawkweed, progeny is produced not after the fusion but before the but by the ovule without fertilization. So they are not having the gametic combination, or we can say the allelic combination. So that's why Mendel failed to show its theory in the hawkweed and the Hieracium. So now we'll understand all these concepts individual. First of all, we'll understand the gene action. So now what is gene action? So basically, gene and allele, as we discussed in the conceptual video, like for a gene of height, we know that there are two allele. One allele is dominant allele, that is denoting for the tallness, or this is the dominant allele, and the second one is dwarf. This is a recessive allele, okay, donating for the dwarfness. So genes control the phenotypic expression. So these are controlling the phenotypic expression, means if this condition is there, so we will call the organism will be tall. If this is the condition, again we will call the, according to the law of dominance, individual will be tall. And if this is the condition, we will call that this individual will be the dwarf according to the combination. So the gene are controlling the phenotypic expression of the various character, not only for the height, for all the characters. Okay, allele of gene may interact with one another in a number of ways. Allele of a gene, these are the two allele, they are interacting in a number of ways, means in all possible combination, although they are making only three type of combinations because only two alleles are there. So alleles of a gene may interact with one another in a number of ways to produce a variability in the phenotypic expression. That's why the variability in phenotypic expression is there. Like this is the variability, one is tall, one is dwarf, and in the incomplete dominance, one is red, one is white, one is pink. So this is the based on the combination of alleles. So the dominant and the recessive relationship is fundamental, and it is essentially constant for all the characters. Now we will study the gene action based on the dominance effect. Now what effect? Dominance effect. Dominance effect means in a gene there will be two allele. One allele will always be dominant, another allele is always be recessive. The gene whose mask is visible in the heterozygote condition is known as a dominant allele. Like capital T and small t, this is a heterozygote condition. So heterozygotic condition whose impact will be visible. So this is a tall plant, so we can say that this allele impact is visible. That's why this is a dominant allele. Now complete dominance. Complete dominance is phenotype produced by the heterozygote will be identical to that produced by the homozygote. This is the condition of complete dominance. Means capital T will lead to the tall plant. Capital T small t will also lead to the tall plant. So this is the concept of complete dominance. Okay, complete dominance. Homozygote is having impact equal to the heterozygote. This is the law. This is the complete dominance condition. Now another condition is incomplete dominance. Incomplete dominance means homozygote is not having the impact similar to the heterozygote. Just take the example of Mirabilis gelapa, as we discussed. Capital R leading to the red flower. Capital R, small r. According to the Mendel law, this should be red flower, but it is showing the pink flower. So this is the deviation from the Mendel's law. That's why we are calling that the incomplete dominance. Means heterozygote is not showing its impact similar to the homozygote. So this is incomplete dominance. The variation in the phenotype of heterozygote. Okay, deviation in heterozygote. Now there is the condition codominance. Codominance means both of the alleles are showing its own impact equally. No one is dominant, no one is recessive. So again, this is the exception to the Mendel's law. Example is the AB blood group. In the AB blood group, both the allele are present, capital IA and the IB, and both are expressing equally. So this is the exception of codominance. But if you will see the A blood group, this is the condition IA or IA small I. But in the AB blood group, IA and IB both are present and both are dominant. So this is the example of codominance. Codominance means both of a allele are of a gene expressive equally in a heterozygotic condition. So AB blood group, not ABO. ABO is the exception to the another condition. We'll see later. So this is the example of codominance. Now the another condition is the overdominance. Overdominance is intensity of character governed by them is greater in the heterozygote than in the two concerned homozygote. Means this is a homozygote, this is another homozygote, heterozygote is lying somewhere this condition. So this is a overdominance, or it may fall into this condition also. You know, any means from the capacity of any of the parent outside to the any of the parent. Then is a pseudo dominance. Pseudo means chota. So the pseudo dominance is expression of a recessive allele in a gene in a hemizygous condition, either due to the sex linkage, that is the color blindness, or due to the chromosomal aberrations. As I discussed in the polyploidy condition, due to the non-disjunction of chromosome, if this is the condition, this is the condition, means, sorry, this is the condition, this is a dwarf allele for a height, or we can say the recessive allele for a height, which is responsible for the dwarfness, and there is no other allele present here. So because there is no other allele, so single allele will able to show its impact. So this is known as the hemizygous condition. In the absence of the dominant allele, recessive allele is showing its impact. So that is known as the pseudo dominance. Okay, so these are the based on the dominance effect. Then based on the lethal effect, lethal as the name suggesting, you know, leads to the death. Dominant lethal means there are some genes affect the survival of those zygote or individual in which they are present in an appropriate genotype, coat colored in mice. Means, just take the example, you know, this is the condition, and the dominant allele is responsible for the lethality. Lethality means for the death of an individual. Means any individual having the capital Y or the dominant condition, they will die, they will be not viable. So these genotypes will not able to survive. Similarly, these genotypes are also not able to survive. Only the recessive, these will be able to survive in the population. So this is the dominant lethal. Then another is the recessive lethal. You know, albino seedling in the albino seedling character in the plant such as rice and barley is governed by the recessive. In this condition, lethality is governed by the recessive allele. So this combination will not viable, and these two combination will be viable, and these two type of individual will survive in the population. So this is the effect of lethal. Then is the based on the epistasis. Epistasis is when expression of one gene depends on presence and absence of another gene in an individual, absolutely deviating the law of independent assortment. In the independent assortment, we were saying that despite of remaining together, you know, during the gamete formation, assortment of one character will be independent of another. But here we are saying presence and absence of one gene is affecting the expression or impression of another gene. So epistatic, so they are basically modifying the F2 ratio. Like this should be the condition 9:3:3:1, and these are the modification to the F2 ratios. Like in the 9:3:3:1 ratio, if we are taking, you know, two type of allele, two type of characters, so this is the condition. Here they may be the dominant allele or this recessive allele. Okay, so the 9 genotype will be like this. Then capital A, small b, 3, then small a, capital B, and here it may be, you know, anyone. Then small a, small b. So 9 is to 3 is to 3 is to 1. This is the perfect phenotype of F2 of the dihybrid cross according to the law of independent assortment or the Mendel's law. But in this condition, 12 is to 3 is to 3, 12 is to 3 is to 1, how it is modifying? If a capital A is present in a dominant condition, so it will resides, or we can say hide the effect of another allele, whether it is present in a dominant condition or it is in the recessive condition. So both will be having the similar impact. So this will modify to 12 is to 3 is to 1. It may be, it may be because of capital B. ये भी हो सकता है capital A and capital B and small a capital B in this condition. So again this will modify to 12 is to 3 is to 1. Similarly the other, 15 is to 1. Any of the dominant allele present in the combination will make the particular type of phenotype. So all these three will make a similar type of phenotype, and small a, small b will make the another type of phenotype. So this is how these are the modification in the due to the epistasis reason, and these are the based on epistatic action. Although these are not very much important, you have to understand the condition of epistasis means one allele is affecting the presence of a particular allele is affecting the expression of another allele, or we can say the gene. So these are the various type of epistasis. So then another type of exceptions we will discuss in our next video, or we can say the next part of this particular series. Other genetic concept or other Mendelian concepts must go through the notes and also attempt the chapter test related to this particular concept. Thank you for watching this video. Hello everyone, welcome you all in this video is DNA structure and its replication. So far we have studied the various concepts of Mendelian genetics, some exceptions to the Mendelian genetics. Now we will study to the molecular genetics side. So this is the very beginning of the molecular genetics. First all of you, we have to understand the DNA structure, what is DNA, what it is made up of, and how the DNA is known to be a genetic material, why DNA is only the genetic material. So we will know all these things in this particular video. Okay, so we'll start with the DNA structure. DNA, what is DNA? First of all, we have to understand what is DNA and what is the nuclear material or the nucleic acid, what is the genetic material. So this will understand. A nucleic acid that carries the genetic information in a cell and that is capable of self-replication is DNA, or it is the molecule present inside the cell that carry the genetic information and pass it to one generation to the next. So in the very basic video of the cell, we have studied that inside the cell there is a nucleus, okay, and inside the nucleus के अंदर क्या होते हैं chromosomes, and chromosomes are nothing but the highly condensed form of the DNA. अगर हम इसको अनवाइंड करते हैं तो यहाँ पे हमको क्या मिलता है DNA. Okay, so double helix standard of DNA, and DNA क्या है, RNA क्या है, क्या उसमें डिफरेंस है वो हम अभी नहीं देखेंगे. तो ये बेसिकली कहाँ पे प्रेजेंट होता है, हमारे न्यूक्लियस में प्रेजेंट होता है किसी भी ऑर्गेनिज्म के, और ये सेल कैपेबल होता है सेल्फ रेप्लिकेशन. नाउ रिमेंबर द सेल साइकिल. सेल साइकिल में क्या हो रहा था कि जो एम फेज था उसके अलावा इंटरफेज था. तो एम फेज में व्हेन सेल स्टार्ट डिवाइडिंग, तो एट द एंड ऑफ एम साइकिल वन सेल इज डिवाइडेड इनटू टू, और यहाँ पे क्या होता है डीएनए मटेरियल हाफ हो जाता है. और ये जो G1, S एंड G2 फेज होते हैं, 3 फेज होते हैं इंटरफेज के, उसमें यहाँ पे इसका जो डीएनए मटेरियल होता है इट गेट डबल. एंड कैसे डबल होता है? बाय द मैकेनिज्म ऑफ डीएनए सेल्फ रेप्लिकेशन. सो दिस वे विल अंडरस्टैंड इन दिस पर्टिकुलर वीडियो. ओके, सो द लोकेशन कहाँ पर है, डीएनए कहाँ पे प्रेजेंट होता है? इट इज इन यूकैरियोट, इट इज प्रेजेंट इन न्यूक्लियस एंड साइटोप्लाज्म आल्सो. हम सभी को पता है देयर आर सम एक्स्ट्रा न्यूक्लियर जेनेटिक मटेरियल इज आल्सो प्रेजेंट, जिसको माइटोकॉन्ड्रियल या फिर जैसे हम साइटोप्लाज्मिक इनहेरिटेंस कहते हैं. व्हाई साइटोप्लाज्मिक इनहेरिटेंस? बिकॉज दोज ऑर्गेनेली अदर देन न्यूक्लियस व्हिच आर प्रेजेंट इन द cytoplasm and responsible for the DNA or we can say the genetic transfer or genetic material. So that's why we use the term cytoplasm, and for the same we use the concept of cytoplasmic inheritance. Like mitochondria also consist of some genetic material, and similarly protoplast. Okay, then in prokaryote it is found in the cytoplasm in a circular form. There is no nucleus and there is no cell bounded organelle present inside the cell. Okay, Then is the structure. If we talk about the structure of DNA, DNA is a double stranded in both prokaryote as well as in the eukaryote. Okay, if there is single stranded, so then it will be known as RNA. Double stranded is DNA. Virus, it is single stranded. In some viruses there is exception that single stranded DNA is also present. Then shape in eukaryote, it is linear, and in prokaryote and mitochondria it is circular in nature. Okay, here in the eukaryote it is circular, linear in nature, and by the condensing of this linear DNA will get the क्रोमोसोम. यहाँ पे क्या होता है, ये लीनियर होता है इस टाइप से, लॉन्ग थ्रेड होगा और इसको कंडेंस करेंगे तो क्रोमोसोम बनेगा. बट प्रोकैरियोट या माइटोकॉन्ड्रिया में क्या होता है, सर्कुलर टाइप का डीएनए प्रेजेंट होता है, वहां पे क्रोमोसोम्स नहीं होते हैं. देन रेप्लिकेशन की अगर हम कैपेबिलिटी की बात करें, इट मस्ट बी ऑफ एबल टू सेल्फ रेप्लिकेट, क्योंकि सेल्फ रेप्लिकेट हो पाएगा तभी वो आगे को जीन ट्रांसफर, या हम कह सकते हैं कि प्रोजेनी जो पेरेंट्स है वहां से प्रोजेनी को ट्रांसफर हो सकता है. नीट रेप्लिकेशन सेमी कंजर्वेटिव मैनर. सेमी कंजर्वेटिव मैनर में क्या क्या मतलब है कि वो हाफ जैसा पेरेंट में है वैसा रहता है, एंड कुछ वो न्यूली सिंथेसाइज करता है स्ट्रैंड्स को. सो दिस इज द सेमी कंजर्वेटिव मेथड. आगे हम पढ़ेंगे इसको. So there are two type of nucleic acid. तो nucleic acid हमने बात की कि nucleic acid is a term used for the genetic material that consists inside the nucleus, or that is responsible for our genetic or the inheritance type of study. Okay, so there are two type of nucleic acid. One is the DNA and other one is the RNA. Okay, so there is the chemical level differences. DNA is the genetic material in most of the organism. RNA acts as a genetic material in some viruses. Okay, so this you have to understand. The DNA is mainly found in the chromosome in the nucleus, while RNA mostly found in the ribosome in the cytoplasm. So this is the component of DNA. DNA is consist of three things. Okay, DNA consists of three things. First is nitrogenous base. Nitrogenous base is again consists of either purines or pyrimidines. Okay, so purines consist of adenine and guanine. Pyrimidine consists of cytosine and thymine. And in the RNA, in case of RNA, instead of thymine, uracil is present. Okay, this we write as A and G, C, T. And according to Chargaff's ruling, whenever the DNA strand will form, there is a base pairing. So pairing of bases between purines and pyrimidine. So A always pair with T and C always pair with G. Okay, so ATCG, this you have to remember. Okay, कि adenine will always pair with the thymine and cytosine will always pair with the guanine. Okay, so these are the complementary bases. And in the RNA, instead of thymine, what is there? Uracil. Okay, so whenever there will be the transcription, means the formation of RNA from DNA. According to the central dogma, so the formation of RNA, so if like there is a sequence like A, T, G, C, like something, so formation of RNA is there. So instead of thymine, the base pairing will consist of uracil, this cytosine, this again adenine, this cytosine, and this guanine. So these will be the base pairing for, or the complementary base pairing for this particular sequence. Okay, so this will study in the further classes. So next is the deoxyribose. So deoxyribose is a pentose sugar having the 5 carbon atoms. This basically combines with the phosphorus and the nitrogenous bases. And third is the phosphates. So the phosphates are help in binding one molecule of nitrogenous bases of deoxyribose sugar plus nitrogenous bases to the another molecule. So this leads to the formation of a chain. So nucleic acid can be broken into a smaller molecule. So by using these three things, there is a one unit. इन तीनों को मिलाकर एक यूनिट बनती है जिसको बार बार रेप्लिकेट करके या बार बार रिपीट करके एक लॉन्ग फॉर्म ऑफ डीएनए बनता है, जिसको हम क्या कहते हैं, न्यूक्लियोटाइड. तो न्यूक्लियोटाइड क्या होती है, राइबोटाइड या फिर डीऑक्सीराइबोटाइड. शुगर प्लस फॉस्फेट ग्रुप प्लस नाइट्रोजनस बेस, दिस इज द न्यूक्लियोटाइड. और न्यूक्लियोसाइड क्या होता है, राइबोसाइड एंड डीऑक्सीराइबोसाइड, दिस इज द शुगर प्लस नाइट्रोजनस बेस. मींस देयर इज नो फॉस्फोरस. तो फॉस्फोरस नहीं है तो मतलब ये आपस में कंबाइन नहीं हो पाएगी, एक यूनिट दूसरी यूनिट से कंबाइन नहीं हो पाएगी. इसीलिए जो आरएनए होता है दैट इज अ सिंगल स्ट्रैंडेड, वो डबल स्ट्रैंडेड नहीं होता है, क्योंकि फॉस्फोरस हमें चाहिए होता है बीच में जो दोनों को अटैच करता है दो डबल स्ट्रैंड्स को. तो वो न्यूक्लियोटाइड्स मिला के बनती है. न्यूक्लियोसाइड्स में केवल शुगर प्लस नाइट्रोजनस बेस होता है. तो न्यूक्लियोटाइड जो होती है इट कंसिस्ट ऑफ न्यूक्लियोसाइड प्लस फॉस्फेट. सो क्या डिफरेंस है न्यूक्लियोसाइड एंड न्यूक्लियोटाइड में, इट इज ओनली ऑफ फॉस्फोरस. नाउ यू कैन सी दिस डायग्राम अगेन. This is the component of DNA. So here you can see that this is the base, or you can say this is the pentose sugar, this is the phosphorus, and this is the nitrogenous base. Okay, so these three elements and these are keep on repeating. Okay, so this leads to the formation of a chain. Okay, so this is the component of DNA. Now you have to understand how the DNA is formed or by these elementary things. So these three commonly known as nucleotide. So if phosphorus is not there, then only these two things. So this will be RNA. Oh, sorry. This will be nucleoside. Okay, nucleotide means addition of phosphorus in the nucleoside. So now we'll study about the DNA as a genetic material with experiment. So this is the first experiment which tried in the search of the genetic material. तो उस टाइम पे क्या था कि पता ही नहीं था कि genetic material क्या है. देखो मेंडल ने उस टाइम पे जब 1966 में अपनी थ्योरी रिप्रेजेंट की थी, उन्होंने कहा था कि there is some factors, and in 1900 में जब रीडिस्कवर हुआ तो वहाँ पे जीन जीनोटाइप एंड फिनोटाइप का कंसेप्ट आया, बट स्टिल इट इज अननोन कि वो कहाँ पे प्रेजेंट है, कैसे ये सारी चीजें होती हैं. तो जो ग्रिफिथ ने एक्सपेरिमेंट किया, तो ग्रिफिथ ने अपने एक्सपेरिमेंट में क्या किया, बेसिकली जो ग्रिफिथ थे वो उनका एक्सपेरिमेंट डीएनए जेनेटिक मटेरियल की खोज करना नहीं था. ही वाज डेवलपिंग अ वैक्सीन फॉर द निमोनिया. तो बेसिकली उनको ये सारी चीजें मिली, तो उन्होंने यहाँ पे क्या यूज किया, यहाँ पे एक स्मूथ स्ट्रेन यूज की जो कि वायरुलेंट थी. स्मूथ स्ट्रेन वायरुलेंट थी. स्मूथ स्ट्रेन क्या थी, मतलब ये जैसे कॉलोनी है, इसके आगे बाहर से एक लेयर थी, तो ये वायरुलेंट थी, मतलब स्मूथ स्ट्रेन थी. और इसी ये जो कैप्सूल प्रेजेंट है, इसी कैप्सूल की वजह से क्या होता था कि जो माइस में जब इंजेक्ट किया जाता था, माइस की एंटीबॉडीज इसको ब्रेक नहीं कर पाती थी और माइस की डेथ हो जाती थी. तो ये वायरुलेंट इन नेचर था कि उन्होंने यूज किया स्मूथ स्ट्रेन. स्मूथ स्ट्रेन जैसे ही माइस में इंजेक्ट किए, और क्योंकि कैप्सूल था, कैप्सूल की वजह से वो उसको जो एंटीबॉडीज थी वो उसको किल नहीं कर पा रही थी, और ये जो स्ट्रेन था ये माइस को किल कर दे रहा था. तो यहाँ पे माउस की डेथ हो गई. रफ स्ट्रेन था तो उसमें कोई भी कैप्सूल नहीं था. कैप्सूल नहीं होने की वजह से जो इसकी एंटीबॉडीज थी वो उसको किल कर दे रही थी, और माउस जो वो जिंदा रह रहा था. देन हीट किल्ड स्मूथ स्ट्रेन. अब उन्होंने क्या किया कि हीट किल्ड स्मूथ स्ट्रेन, जो कैप्सुलेटेड थे उनको हीट किल कर दिया. अब हीट किल कर दिया मतलब वो एक्टिव नहीं है, तो जब उसको इंजेक्ट किया तो माउस जिंदा रहा. बट एक स्ट्रेन जो रिजल्ट यहाँ पे मिला, इससे ये सारी चीजें चेंज हुई. इसमें इन्होंने रफ स्ट्रेन को यूज किया और उसके साथ ही साथ हीट किल्ड स्मूथ स्ट्रेन. तो आइडियली क्या होना चाहिए था, जब सिंगल कंडीशन थी कि रफ स्ट्रेन में भी माउस जिंदा था और हीट किल्ड स्मूथ स्ट्रेन थी जो उसमें भी माउस जिंदा था, बट यहाँ पे माउस की डेथ हो गई. तो उन्होंने कहा कि देयर इज समथिंग विच इज रेप्लिकेटिंग या फिर विच इज ट्रांसफॉर्मिंग, इसीलिए इसको ट्रांसफॉर्मिंग प्रिंसिपल कहते हैं हम लोग. ये उन्होंने कहा कि देयर इज समथिंग विच इज ट्रांसफॉर्मिंग. तो उसमें क्या हो रहा था कि हीट किल्ड जो स्मूथ स्ट्रेन था, जो स्मूथ का स्ट्रेन था वो कुछ ना कुछ चेंज हो रहा है, जो ट्रांसफॉर्म कर रहा है, जिसकी वजह से ये माउस की डेथ हुई. बट वो बता नहीं पाई ये सारी चीजें कि ये कैसे हुआ. तो बेसिकली द केमिकल फ्रॉम द हीट किल्ड बैक्टीरिया ट्रांसफॉर्म द ए वायरुलेंट बैक्टीरिया इनटू वायरुलेंट. तो उसने जो ए वायरुलेंट स्ट्रेन था रफ का, उसको वायरुलेंट कर दिया. हाउएवर ग्रिफिथ कुड नॉट कंक्लूड दैट द ट्रांसफॉर्मिंग केमिकल वाज डीएनए. तो वो नहीं बता पाए कि जो ये ट्रांसफॉर्मिंग मटेरियल है ये डीएनए है. ओके, तो ये खोज आगे चलती रही. Then next second attempt was done by the Avery McCarty and MacLeod experiment. हालांकि इसके बाद ये कंफर्म हो गया था, बट अगेन जो फाइनल रिजल्ट थे वो बाद में आए थे. तो डीएनए वाज जेनेटिक मटेरियल बट नॉट प्रोटीन. ये उन्होंने बताया कि डीएनए जेनेटिक मटेरियल है, प्रोटीन नहीं है, क्योंकि जब ट्रांसफॉर्मिंग प्रिंसिपल की बात आई तो कुछ भी ट्रांसफॉर्म हो सकता है. प्रोटीन भी हो सकता है, आरएनए भी हो सकता है, डीएनए भी हो सकता है. तो अगेन ये पता नहीं था. तो जो एवरी मैककार्टी एंड मैक्लियोड के एक्सपेरिमेंट से इट वाज क्लियर दैट डीएनए वाज द जेनेटिक मटेरियल. अब इन्होंने क्या किया, जो एस टाइप स्ट्रेन था, स्मूथ स्ट्रेन था, उसको हीट किल कर दिया. हीट किल करके उसकी कॉलोनी को बना दिया यहाँ पे. ठीक है, तो ये आप देखोगे तो ये पूरा स्ट्रक्चर यही है कि इन्होंने हीट किल को उसकी कॉलोनीज बना ली. हीट किल्ड कॉलोनीज में अब क्या डाउट है कि क्या पता प्रोटीन हो सकता है, या तो आरएनए हो सकता है, या तो डीएनए हो सकता है. उन्होंने सिंपल फॉर्मूला यूज किया कि अगर हम इसमें प्रोटीएज डाल देंगे, प्रोटीएज क्या एंजाइम है जो कि प्रोटीन को डिग्रेड कर देगा. प्रोटीन को डिग्रेड कर देगा तो मतलब वो जो वायरुलेंस है वो खत्म हो जाएगी, और जो हमको वो वाली थर्ड एस की कॉलोनीज नहीं मिलेंगी. तो यहाँ पे इन्होंने क्या किया, प्रोटीएज से ट्रीट किया एस टाइप को, और यहाँ पे इन्होंने सेकंड आर इसमें डाला और प्रोटीएज ट्रीटेड थर्ड एस कॉलोनीज. तो यहाँ पे आर भी मिला, इनको एस भी मिला. तो इसका क्या मतलब है कि जो प्रोटीएज था उसने जिसको भी डिग्रेड किया वो जेनेटिक मटेरियल नहीं है, क्योंकि यहाँ पे हमको कॉलोनीज मिल गई। तो ये भी यहाँ पे क्लियर हो गया कि प्रोटीन इज नॉट द जेनेटिक मटेरियल। देन इन्होंने राइबोन्यूक्लियस से ट्रीट किया कि ये डाउट है कि अगर आरएनए है तो ये आरएनए को ट्रीट कर दे तो आरएनए की कॉलोनी हमें नहीं मिलेंगी। तो अगेन सेम चीज रिपीट की गई, सेकंड आर को और आरएनए इज ट्रीटेड, थर्ड एज फिल्ट्रेट। और ये थर्ड एज फिल्ट्रेट कैसा है, वो हीट किल्ड है, जैसा हमारा लास्ट वाला था जो ट्रांसफॉर्मिंग प्रिंसिपल ग्रिफिथ नहीं बता पाए थे। अगेन जब यहाँ पे देखा गया तो सेकंड आर और एस कॉलोनी मतलब रफ एंड स्मूथ दोनों स्ट्रेट पाए गए। इसका मतलब क्या था कि अगेन जो आरएनए है उसने आरएनए को डिग्रेड किया बट स्टिल वो प्रेजेंट है। इसका मतलब कि आरएनए भी जेनेटिक मटेरियल नहीं है। देन थर्ड अटेम्प्ट में किया गया डीऑक्सीराइबोन्यूक्लियस से ट्रीट किया गया जो डीएनए को ब्रेक कर देगा। तो देन अगेन सेकंड आर प्लस डीएनए इज ट्रीटेड, थर्ड एज फिल्ट्रेट को केवल यहाँ पे सेकंड आर कॉलोनीज मिली, थर्ड एज कॉलोनीज नहीं मिली। तो इससे कंफर्म हो गया कि दैट जेनेटिक मटेरियल जो यहाँ पे ट्रांसफॉर्म कर रहा है, जो चेंज हो रहा है वो क्या है, डीएनए। क्यों? क्योंकि यहाँ पे डीएनए से पहले ट्रीट कर दिया हमने डीएनए से, तो डीएनए से जब ट्रीट करेंगे तो डीएनए को खत्म कर देगा। तो डीएनए को खत्म कर दिया मतलब वो नहीं मिला, तो इसका मतलब है कि जो ट्रांसफॉर्म कर रहा था वो क्या था, डीएनए था। सो इट वाज क्लियर बाय द एवरी मैककार्टी एंड मैक्लियोर। तो इन्होंने बताया कि डीएनए वाज अ जेनेटिक मटेरियल नॉट प्रोटीन बिकॉज़ द ट्रांसफॉर्मेशन अकर्ड व्हेन डीएनए वाज प्रेजेंट इन द एक्सट्रैक्ट एंड देयर इज नो ट्रांसफॉर्मेशन व्हेन डीएनए वाज डाइजेस्टेड बाय डीएनएज। तो डीएनएज ने जब डाइजेस्ट किया उसके बाद कोई ट्रांसफॉर्मेशन नहीं हुआ, बट जब तक डीएनएज नहीं था तब तक ट्रांसफॉर्म हो रहा था, और वो इसीलिए हो रहा था क्योंकि डीएनए था। अब अगर आपने प्रोटीन को ट्रीट कर भी दिया तो डीएनए ट्रांसफॉर्म होगा, आरएनए से ट्रीट कर भी दिया तब भी डीएनए ट्रांसफॉर्म होगा। जब डीएनए से ट्रीट कर दिया तब वो ट्रांसफॉर्म नहीं हो पाएगा। इसलिए हमारा ये क्लियर हुआ कि यहाँ पे डीएनए इज द जेनेटिक मटेरियल या डीएनए इज द ट्रांसफॉर्मिंग एलिमेंट। Okay, so this was the experiment by the Avery, McCarty and MacLeod in the 1944. Then final experiment, Hershey and Chase experiment 1951 and 52। तो इन्होंने फाइनल कंक्लूड किया, they did the simple experiment। इन्होंने क्या किया, बैक्टीरियोफेज को लिया। बैक्टीरियोफेज क्या होते हैं, these are the phages which infect the bacteria। या फिर कह सकते हैं टाइप से इसको वायरस भी कह सकते हैं कि ये किसको इन्फेक्ट करते हैं, बैक्टीरिया को। तो इसमें क्या होता है, इनका बहुत सिंपल स्ट्रक्चर होता है। एक तो इनमें प्रोटीन कोट होता है और दूसरा न्यूक्लिक एसिड होता है, यहाँ पे राउंड एंड सर्कुलर। तो यहाँ पे इन्होंने दो एक्सपेरिमेंट किए। पहले में प्रोटीन कोट था, इसको रेडियो एक्टिव लेबल कर दिया, सो दैट कि जैसे ही वो इन्फेक्ट करेगा तो हमें पता चल जाएगा रेडियो एक्टिविटी से कि जहाँ भी रेडियो एक्टिव शो हो रही है वो जेनेटिक मटेरियल है। तो इन्होंने प्रोटीन कोट को रेडियो एक्टिव बना दिया। दूसरे वाले में इन्होंने जो डीएनए था फेज का उसको रेडियो एक्टिव बनाया। अब जैसे ही इन्होंने बैक्टीरिया में इन्फेक्शन कराया यहाँ पे, तो जैसे इन्फेक्शन कराया तो क्योंकि उस टाइम पे ये क्लियर नहीं था, आज के टाइम पे हमें पता है कि डीएनए ही अंदर जाता है। तो वहां पे जैसे ही ये मटेरियल अंदर गया ये बाहर छूट गए और कोई रेडियो एक्टिविटी नहीं दिखी। रेडियो एक्टिविटी क्यों नहीं दिखी, क्योंकि रेडियो एक्टिविटी किसमें थी, प्रोटीन में। प्रोटीन कोड तो पार्टिसिपेट ही नहीं करता ट्रांसफॉर्मिंग में। तो जब दूसरे एक्सपेरिमेंट में जिसमें डीएनए को रेडियो लेबल किया गया था, रेडियो एक्टिविटी इसमें शो की गई थी, तो जब यहाँ पे इसको इन्फेक्ट किया गया तो यहाँ पे देखा कि रेडियो एक्टिविटी एंटर्स द सेल। क्यों? क्योंकि जो डीएनए था वो ट्रांसफर हुआ, प्रोटीन को ट्रांसफर नहीं होते। तो इससे क्लियर हो गया था कि जो रेडियो एक्टिव जो डीएनए है दैट इज द ट्रांसफॉर्मिंग प्रिंसिपल या ट्रांसफॉर्मिंग एलिमेंट। तो इन्होंने कंक्लूड किया ओनली द फेजेस व्हिच आर लेबल्ड विद द रेडियोएक्टिव फास्फोरस P32 transmitted radioactivity in the daughter phases. This provided a strong evidence that DNA is the genetic material, not the protein. तो यहाँ से क्लियर हो गया था कि DNA is the genetic material, not the protein. They concluded that genetic material must be DNA carrying radioactivity inside the bacteria and to the offspring phages। तो यहाँ से फाइनली क्लियर हो गया था कि the DNA is the genetic material, not the protein। Then अब हम पढ़ेंगे डीएनए रेप्लिकेशन के बारे में। सो अब हमको क्लियर हो गया कि डीएनए इज द जेनेटिक मटेरियल। नाउ डीएनए रेप्लिकेशन। अभी हमने कहा था कि जेनेटिक मटेरियल हम किसे कहते हैं, कि जो जिसमें कैपेसिटी होगी सेल्फ रेप्लिकेशन की। अब वो सेल्फ रेप्लिकेशन क्या है और सेल्फ रेप्लिकेशन क्यों जरूरी है। सो यू विल गेट दिस आंसर बाय द टाइम ऑफ रेप्लिकेशन। डीएनए रेप्लिकेशन कब होता है, ड्यूरिंग द सेल डिवीजन प्रिसाइजली एज सब स्टेज ऑफ इंटरफेज। तो अभी मैंने जैसे आपको बताया था कि ये सेल साइकिल है, ये इंटरफेज है, ये एम साइकिल है। ओके, तो यहाँ पे सेल स्टार्ट हुआ डिवाइड होने, यहाँ पे 1 से 2 सेल बन गए, तो मतलब जो दोनों न्यूक्लियर मटेरियल था वो हाफ हो गया। अब ये पूरी इंटरफेज में तीनों साइकिल में G1 S एंड G2 में ये अगेन अपना जो न्यूक्लियर मटेरियल है उसको बनाएगा, मतलब डीएनए को रेप्लिकेट करेगा। डीएनए हाफ हो गया तो उसको रेप्लिकेट करेगा। सो दिस इज द टाइम ऑफ रेप्लिकेशन। देन रेप्लिकेशन साइट क्या होती है, कहां पे ये रेप्लिकेट होगा, तो यूकैरियोट में सेल न्यूक्लियस में, क्योंकि क्रोमोसोम या डीएनए कहां प्रेजेंट है, सेल न्यूक्लियस में। प्रोकैरियोट में वो क्योंकि साइटोप्लाज्म है तो वो साइटोप्लाज्म में ही होगा। टेम्पलेट यूज्ड, टेम्पलेट क्या होता है कि बेस कहां से वो यूज करेगा, तो जो एग्जिस्टिंग डीएनए होती है उसको ही टेम्पलेट यूज किया जाता है फॉर द सिंथेसिस। अब इसकी डिटेल्स हम आगे डीएनए रेप्लिकेशन जब पूरा मॉडल पढ़ेंगे तो उसमें हम डिस्कस करेंगे। 5 days 3 days 3 days 5 days वो पूरा उसमें पढ़ेंगे। न्यू स्ट्रैंड ऑफ डीएनए दिस आल्सो कंटीन्यूअस रेप्लिकेटिंग स्ट्रैंड। होता क्या है कि हमारी दो स्ट्रैंड होती हैं, आपने देखा होगा कि रेप्लिकेशन फोक जब भी बनता है ये 3 dash टू 5 dash है। अभी 3 dash 5 dash का क्या मतलब है ये हम अपनी उस वाली वीडियो में देखेंगे। देन ये 5 dash टू 3 dash, तो यहाँ से रेप्लिकेशन स्टार्ट होता है इस टाइप को। और ये क्या होता है, कंटिन्यूअस स्ट्रैंड होता है तो इसको हम लीडिंग स्ट्रैंड बोलते हैं। यहाँ पे डिसकंटिन्यूअस होता है तो इसको हम क्या बोलते हैं, लैगिंग स्ट्रैंड। क्योंकि ये जो डायरेक्शन होता है ये 3 to 5 ही होती है हमेशा, तो यहाँ पे जो फॉर्म बनेगी वो कैसे बनेगी 3 to 5। अब ये फोक ऊपर को ओपन होता जाता है, ऊपर को जैसे ही ओपन होगा तो ये तो कंटिन्यूअस बनता रहेगा, बट यहाँ से बनना शुरू हो गया था तो अगेन इसलिए ये डिसकंटिन्यूअस होता है। क्योंकि ये फोक ऊपर को खुल रहा है, जैसे ही फोक ऊपर ओपन होगा तो नीचे को सिंथेसिस करनी पड़ती है, तो इसलिए ये डिसकंटिन्यूअस होता है। कई स्टूडेंट्स को डाउट होता है कि ये डिसकंटिन्यूअस क्यों है, ये कंटिन्यूअस क्यों है, ये भी तो कंटिन्यूअस बन सकता है। तो इसलिए क्योंकि फोर्क ओपन ही नहीं है। अभी इतना फोर्क ओपन है तो ये तो कंटिन्यूअस ऊपर को बना रहा है, आगे भी ओपन होता जाएगा तो ये बनाता जाएगा। बट यहाँ पे क्या है, अभी इतना फोर्क ओपन था उसने यहाँ से बनाना इधर को शुरू किया, बट अब जब आगे और ओपन हो गया तो फिर ये तो यहीं से शुरू होता है, ये तो इधर को बना रहा था, तो ऊपर के लिए दोबारा से एक स्टैंड बनती है। तो इसलिए ये लैगिंग स्टैंड या फिर डिसकंटिन्यूअस स्टैंड हम इसको कहते हैं। ये हम डीएनए रेप्लिकेशन में कंप्लीटली पढ़ेंगे, इसको भी यहाँ पे डिस्कस नहीं करते। और ये जो छोटे छोटे फ्रेगमेंट्स बनते हैं ये ओकाजाकी साइंटिस्ट ने डिस्कवर किए थे, इसलिए इसको हम ओकाजाकी फ्रेगमेंट्स कहते हैं। एंजाइम कौन से इन्वॉल्व होता है, डीएनए पॉलीमरेज, क्योंकि डीएनए बनना है। देन सेमी कंजर्वेटिव मेथड ऑफ द डीएनए रेप्लिकेशन। अब आता है कि ये क्या चीज है सेमी कंजर्वेटिव, तो इसमें क्या होता है, डीएनए रेप्लिकेशन ईच न्यूली सिंथेसाइज्ड स्ट्रैंड ऑफ डीएनए वुड रिमेन एसोसिएटेड विद द ओल्ड स्ट्रैंड अगेंस्ट व्हिच इट वाज सिंथेसाइज्ड। तो एक स्ट्रैंड पुरानी होगी और एक स्ट्रैंड नई होगी। जैसे अभी हमने देखा कि ये डीएनए रेप्लिकेशन है तो एक से दो बनेगा, तो यहाँ पे ये कंटिन्यूअस स्ट्रैंड बन रही है, ये दूसरी स्ट्रैंड, तो ये एक पूरा बन जाएगा और ये दूसरा बन जाएगा। तो इस टाइप से डीएनए रेप्लिकेट होता है। सो दस ईच प्रोजेनी डीएनए मॉलिक्यूल कंसिस्ट ऑफ वन ओल्ड एंड वन न्यूली सिंथेसाइज्ड। और इसी वजह से वेरिएशन आते हैं, जो पेरेंट्स ऑफस्प्रिंग में वेरिएशन आते हैं इसी वजह से आते हैं। अगर वो सिमिलर होती, पूरा का पूरा ओल्ड होता, तो उसमें कोई भी वेरिएशन नहीं आता। तो वेरिएशन का रीजन भी क्या होता है प्रोजेनी में, कि डीएनए एक स्ट्रैंड पुरानी है और एक स्ट्रैंड नई है। सो एविडेंस फॉर द सेमी कंजर्वेटिव रेप्लिकेशन ऑफ डीएनए किसने दिया था, मीसलसन एंड स्टॉल ने 1958 में। ई कोलाई में उन्होंने स्टडी की थी और उन्होंने दिया था कि जो ये सेमी कंजर्वेटिव मेथड से रेप्लिकेट होता है। अब ये एक्सपेरिमेंट था क्या, उन्होंने क्या किया, अगेन जैसे ये उसमें हमने रेडियो एक्टिविटी यूज की थी, तो यहाँ पे इन्होंने आइसोटोप से लेबल कर दिया नाइट्रोजन को। तो यहाँ पे इन्होंने क्या किया, 15N डीएनए लिया और उसको किस में मिक्स किया, 14N डीएनए में। और इसके साथ में ये इसको सेंट्रीफ्यूज कर रहे थे। तो जो नाइट्रोजन का 15 आइसोटोप था वो हैवी है, तो वो कहाँ पे सेटल होगा, बॉटम में, और जो N14 है वो लाइट है तो वो ऊपर सेटल होगा। तो उन्होंने क्या किया कि N15 डीएनए को ग्रुप रेप्लिकेट किस में किया गया, N14 डीएनए में। तो तब हमें पता नहीं था कि एक स्ट्रैंड पेरेंट की होती है, एक स्ट्रैंड नहीं बनती है। तो उन्होंने यहाँ पे देखा कि जब ये बनाया तो उनको एक स्ट्रैंड तो पुरानी मिली और एक स्ट्रैंड नहीं मिली। तो ये कैसे पता चला कि जब उन्होंने सेंट्रीफ्यूज किया, क्योंकि देख तो सकते नहीं थे, तो जब सेंट्रीफ्यूज किया तो ये कहाँ पे आया, मिडिल में, जबकि जो पेरेंट था वो कहाँ पे था, बॉटम में। बॉटम में क्यों था कि N15 कैसा था, हैवी। अभी ये मिक्स हो गया, मतलब ना तो ये हल्का है ना तो ये भारी है, तो ये कहाँ पे मिडिल में इसने लाइट बनाई। जब अभी ये बिल्कुल हाइब्रिड है कि इसमें एक स्ट्रैंड N15 की, एक स्ट्रैंड N14 में, दूसरे में भी एक स्ट्रैंड N15 की, N14 की है। यहाँ पे क्या हुआ, जब इसको एक दोबारा सेकेंड जनरेशन में रेप्लिकेट कराया गया, तो जो दो स्ट्रैंड थी, तो एक जो N14 की थी और N14 ही मीडियम है, तो कंप्लीट कौन सी बन जाएगी, N14 की। अब जो N15 वाली थी, N14 मीडियम है, तो ये अगेन हाइब्रिड टाइप हो गई, ये भी हाइब्रिड टाइप हो गई। और यहाँ पे जो N15 वाली थी ये प्योर टाइप N14। तो हमको दो टाइप के स्ट्रैंड मिलेंगे डीएनए के। वो कैसे मिलेंगे, एक तो कंप्लीट N14 टाइप। तो N14 कैसा आइसोटोप था, लाइट आइसोटोप था, तो वो कहाँ पे सेटल होगा, सेंट्रीफ्यूज में ऊपर सेटल होगा। और जो ये हाइब्रिड टाइप थे N15 और N14 के, दोनों में एक एक स्ट्रैंड थे, तो वो मिडिल में सेंट्रीफ्यूज होगा। तो इससे पता चला कि जो रेप्लिकेशन है वो सेमी कंजर्वेटिव है, मतलब एक स्ट्रैंड इंटैक्ट रहती है पेरेंट की और दूसरी नहीं बनती है। तो इससे पता चला कि दिस सेमी कंजर्वेटिव मेथड से डीएनए रेप्लिकेट
होता है और इसी वजह से वेरिएशन आते हैं, वो सारी चीजें थी। तो दिस इज अबाउट द डीएनए एंड इट्स रेप्लिकेशन। कंप्लीट रेप्लिकेशन क्या होता है वो हम नेक्स्ट वीडियो में पढ़ेंगे। देन विल स्टार्ट आवर मॉलिक्यूलर जेनेटिक्स।